Bismillahirrahmanirrahim. Assalamu alaikum, dear cadets. Our topic is mechanism or mode of enzyme action. Before moving into the mechanism or mode of enzyme action, I will explain the reaction mechanism. As you earlier studied in previous class, that in an enzyme catalyzed reaction, the substrate first binds to the active site of the enzyme to form an enzyme substrate complex. Then the substrate is converted into product while attached to the enzyme known as enzyme product complex, and finally the product is released. The enzyme is then free to start another reaction again. Now if you look into this diagram, so the substrate molecule first binds to the active site of the enzyme, substrate entering the active site of the enzyme, forming an enzyme substrate complex. Then the substrate is converted into product. Now the substrate is changed into product while still attached to the enzyme. This time it is known as enzyme product complex, and finally the product is released and the enzyme is free to start another reaction again. This is how the enzyme works. Now coming into the mechanism of enzyme action, so there are two hypotheses which explain the mode of enzyme action. One is known as lock and key model and the other is known as induced fit model. First we will study about lock and key model. The lock and key model was proposed by a German chemist Emil Fischer in 1990. According to this model, as per a specific lock, there is a specific key. Similarly, per a specific substrate, there is a specific enzyme. According to this hypothesis, the substrate is imagined like a lock while the enzyme is imagined like a key. As one specific key can open only a specific lock, similarly a specific enzyme can break up only one specific substrate. According to this model, the active site is a rigid structure that is not flexible, which means the active site does not modify or change during the reaction process. However, later studies or research did not support this hypothesis in all types of reactions, and therefore this hypothesis was modified into induced fit model or hypothesis. Now we will study induced fit model, which was proposed by an American biologist Daniel Koshland in 1959. According to this model, the active site is not a rigid structure and suggested that when a substrate combines with enzyme, it stimulates or induces changes in the enzyme structure, especially at its active site. So this change helps an enzyme to perform its catalytic activity more effectively. This can be imagined like a hand and glove relationship. The substrate, as the hand changes the shape of the glove as it is put on, so the hand is imagined like a substrate while the glove is imagined like the active site of the enzyme. So this is all about the mode or mechanism of enzyme action and the two hypotheses, lock and key model and induced fit model. Thank you.